Dear friends, it's hearty and warm welcome once again. Dear students, in this lecture, we people are going to study two different topics. First, genomics, and second, human genome project. So first of all, let us talk about genomics. The term genomics, first of all coined by scientist T.H. Roderick in year 1986. First of all, let me define what is genomics. Very simple. Genomics, it refers to the study of genome of living organism. Genomics, it refers to the study of genome of living organism, or genomics, it refers to the study of genome through analysis of different genes present in the genome. Analysis in the sense the sequencing and mapping of all the different genes present in the genome, and also it refers to the study of functional aspect of all the genes present in the genome. So we can define genomics in this manner as genomics is the study of genome through analysis of all the genes present in that genome, through mapping and sequencing of all the genes present in the genome, and also the study of functions of all the genes. Now first of all question arises, what does it mean genome? सुरुवातीला आपण जिनॉमिक्स म्हणजे काय याचा अर्थ समजून घेत होतो. मी आता असं म्हटलं की जिनॉमिक्स इज नथिंग बट द स्टडी ऑफ जिनोम. जिनॉमिक्स याचा अर्थ जिनोमचा स्टडी. पण जिनोम म्हणजे काय? व्हेरी सिंपल. जिनोम रिफर्स टू द कंप्लीट और एंटायर जेनेटिक कॉम्प्लिमेंट, एंटायर जेनेटिक कॉन्स्टिट्यूशन, or the entire genetic information present in genes of single set of chromosome, or the genome, it refers to the entire genetic constitution, entire genetic complement present in the single set of chromosomes, that is nothing but the haploid set of chromosome. याचा अर्थ हॅप्लॉइड सेट ऑफ क्रोमोझोम्स मध्ये असणारे जेवढे जीन्स आहेत त्यांना एकत्रितपणे त्या सजीवाचं जिनोम असं रिफर केलं जातं. आणि या एका जिनोम मध्ये असणारे जेवढे जीन्स आहेत त्या सगळ्यांचं मॅपिंग, सिक्वेन्सिंग अर्थात त्यांचं ॲनालिसिस, त्यांचं कार्य, त्यांचं फंक्शनल रोल, हा सगळा अभ्यास बायोलॉजीच्या ज्या डिसिप्लिन मध्ये केला जातो त्याला म्हटलं जातं जिनॉमिक्स. सो आय एम गोइंग टू रिपीट द टू डेफिनेशन वन्स अगेन. जिनॉमिक्स इट रिफर्स टू द स्टडी ऑफ जिनोम थ्रू द एनालिसिस ऑफ जिनोम, थ्रू द सिक्वेन्सिंग मॅपिंग ऑफ जीन्स प्रेझेंट इन द जिनोम, while genome it refers to the entire genetic constitution, entire genetic complement present in the single set of chromosome present in the haploid set of chromosome. आता या ठिकाणी मी दोन शब्द वापरले की जिनॉमिक्स मध्ये सिक्वेन्सिंग अँड मॅपिंग ऑफ जीन्स इज कॅरीड आउट. Sequencing and mapping of genes present in the genome is carried out. Now, what is sequencing and what is mapping? Mapping of genes म्हणजे काय? Mapping of genes it describes the location or locus of a gene in that genome. विशिष्ट प्रकारचा जीन हा त्या सजीवाच्या जिनोम मध्ये कुठे आहे, त्याचं लोकेशन कुठे आहे, त्याचं लोकस कुठे आहे, कुठे पोझिशन आहे, याला म्हटलं जातं मॅपिंग. आणि सिक्वेन्सिंग याचा अर्थ इट रिफर्स टू द स्टडी ऑफ न्यूक्लियोटाइड सिक्वेन्स or the order of nucleotides of that DNA segment, that is nothing but the gene. Because after all we are known what is gene, a particular segment of DNA. मग त्या जीन मध्ये असणाऱ्या न्यूक्लिओटाइडची ऑर्डर काय, ॲडेनिन, ग्वानिन, थायमिन, सायटोसिन, याच्या अभ्यासाला म्हटलं जातं सिक्वेन्सिंग ऑफ जीन अँड द स्टडी ऑफ लोकेशन, स्टडी ऑफ लोकस ऑफ अ पर्टिक्युलर जीन इन दॅट जीनोम इज नोन ॲज द मॅपिंग. And the study of mapping and sequencing of all the genes present in a single or haploid set of chromosome is nothing but the genomics. Now, the genomics, it is mainly divided into two types. Genomics चे दोन प्रकार पाडले जातात, such as structural genomics and functional genomics. Genomics चे दोन प्रकार आहेत, structural and functional. Structural genomics, it refers to the study of mapping, sequencing of genes present in the gene. Or structural genomics, it refers to the study of structure of proteins encoded by the genome. Structure of proteins encoded by the genome. याचा अर्थ त्या जीनोम मध्ये असणारे जे जीन्स आहेत ते जीन्स ज्या प्रोटीन्सला एन्कोड करतात, ज्या प्रोटीन्सचं इन्फॉर्मेशन त्यांच्यामध्ये आहे, अर्थात त्या प्रोटीन्सची स्ट्रक्चर कसं आहे याच्या अभ्यासाला म्हटलं जातं स्ट्रक्चरल जिनोमिक्स. इट मीन्स द स्ट्रक्चरल जिनोमिक्स इट रिफर्स टू द सिक्वेन्सिंग अँड मॅपिंग ऑफ जीन्स or the study of structure of proteins encoded by the genes present in that genome. While functional genomics, it refers to the study of functions of genes present in that genome or function of proteins encoded by the genes present in that genome. म्हणजे functional genomics मध्ये जीनोम मध्ये असणारे जीन्स ज्या प्रोटीन्सला एन्कोड करतात त्यांचं कार्य काय आहे याच्या अभ्यासाला म्हटलं जातं फंक्शनल जिनोमिक्स. नाउ द सायंटिस्ट परफॉर्म द स्टडी ऑफ जीनोम ऑफ डिफरंट लिव्हिंग ऑर्गॅनिझम सच as unicellular fungus, that is yeast, a small insect, that is Drosophila melanogaster, fruit fly. Also perform the study of genome of mouse just to compare the genome of that different living organisms with the genome of human being. It means for the comparative study of the genomes of different living organisms, here the study of genome were carried out of different living organisms, and here through that study so many different amazing facts came into light. शास्त्रज्ञांनी वेगवेगळ्या सजीवांचा तुलनात्मक अभ्यास करण्यासाठी किंबहुना ह्यूमन जिनोमचा आणि इतर सजीवांच्या जिनोमचा कंपॅरेटिव्ह स्टडी करण्यासाठी, तुलनात्मक अभ्यास करण्यासाठी इतर अनेक वेगवेगळ्या सजीवांच्या जिनोमचा अभ्यास केला. आणि हा करत असताना काही अतिशय अमेझिंग अशा गोष्टी समोर आल्या. Here I have mentioned here at the blackboard important facts related with the genome of yeast, fruit fly, mouse and human being. So let us consider, let us talk about it. First of all, let us talk about the yeast. You know, Saccharomyces cerevisiae. Yeast, it is an example of unicellular fungus. It is unicellular fungus. In yeast, the total number of chromosomes, that is diploid number of chromosomes, are 32, while the estimated number of genes are 6,000. Estimated number of genes are 6,000 and the estimated size of genome, of course, two sets of genome, that is 12 million. It means there are presence of 12 million base pairs. It is also called as the C value. One thing I want to tell you, as we have studied genome it refers to the entire set of genes present in single set of chromosome, haploid set of chromosome, and therefore in prokaryotic organisms like bacteria, आपल्याला माहिती आहे की बॅक्टेरिया हे प्रोकॅरियोटिक आहे, and in prokaryotic organism there is a presence of only single chromosome, right? It means the bacteria, prokaryotic organisms are haploid. In such haploid or prokaryotic organisms like bacteria, only one genome is present. Okay? While in eukaryotic cells, as all somatic cells, all general body cells are diploid, it means in such diploid eukaryotic cells there are presence of two genomes, while in polyploid cells, polyploid means the cells in which many sets of chromosomes are present, triploid, tetraploid, pentaploid, whatever it may be, there is number of genomes, अनेक genomes असतात. म्हणजे ट्रिप्लॉइड सेल मध्ये 3 genomes, टेट्राप्लॉइड सेल मध्ये 4 genomes, पेंटाप्लॉइड मध्ये 5 genomes, and so on. It means in haploid prokaryotic bacterial cell there is a single genome. In diploid eukaryotic cells there are 2 genomes, while in polyploid cells there are presence of many genomes. Here we were discussing about the important facts seen in these different living organisms. Yeast, number of chromosome 32, of course diploid, estimated gene number 6000, and base pairs 12 million. Base pairs, it also known as the C value. So the C value of yeast is 12 million, while in fruit fly, a small creature, small insect, scientific name Drosophila melanogaster, the total number of chromosome, total diploid number of chromosomes are only 8, while the estimated number of genes are 13,000. You just compare the number of chromosomes in yeast, number of chromosomes in fruit fly, and also the number of genes in yeast, number of genes in fruit fly. Here in fruit fly, number of base pairs C value is about 16.5 million. 16.5 million. In mouse, scientific name Mus muscularis. You know mouse, it is a mammal. In mouse, there are presence of 20 pairs of chromosome. That is the total number of chromosome in diploid cell 40. The number of genes are 25,000, while the C value, number of base pairs are about 2.9 billion. And in human being, the total number of chromosomes in diploid cell are 46, that is 23 pairs. And these 23 pairs of chromosome contain near about 33,000 genes, and the C value of human being is 3 billion. C value in the sense the total number of base pairs which form the genome. Okay. Now here we have understood about the important facts of all these 4 different living organisms. Now let us talk briefly about the applications of genomics. Genomics application काय आहे? Genomicsचा प्रत्यक्ष कोणकोणत्या क्षेत्रामध्ये उपयोग करून घेतला जातो ते आपल्याला या ठिकाणी शिकायचं आहे. Let us talk about it. The knowledge and understanding of genomics is helpful in so many different fields such as medicine, biotechnology, agriculture, plant breeding, animal breeding, in forensic science, etc., etc. So let us talk few of them applications. First, the knowledge of genomics enhances the basic understanding of human genetics. the knowledge of genomics enhances, means it has to improve our knowledge, it has to increase our knowledge about the human genetics. Second, the genomics, it reveals the genes responsible to develop different genetic disorders. Genetic disorders in the sense hereditary diseases, hereditary disorders like Alzheimer's disease, cardiovascular diseases, cancer, diabetes, etc. वेगवेगळे अनुवंशिक रोग घडवून आणणारे जे जीन्स आहेत त्यांच्या संदर्भातली माहिती जिनॉमिक्सच्या अभ्यासातून मिळू शकते. अँड ऑल्सो द नॉलेज ऑफ जिनॉमिक्स इट इज हेल्पफुल हाऊ टू प्रिव्हेंट these different genetic disorders. So here I have written the genomics, it provides information to prevent such disorders. How to prevent these genetic disorders and also how to treat these genetic disorders by using a special technique known as gene therapy. फक्त genomicsच्या स्टडी मधून कोणकोणत्या जीन्समुळे कोणकोणत्या जेनेटिक डिसऑर्डर्स, अनुवंशिक डिसऑर्डर्स, अनुवंशिक आजार हे निर्माण होता एवढं समजत नाही, तर त्याच्या मदतीने हे आजार कसे टाळले जावेत किंवा त्यांना कशा प्रकारे ट्रीट केलं जावं, जीन थेरपी या टेक्निकचा उपयोग करून याचं सुद्धा नॉलेज जिनॉमिक्स मधून मिळू शकतं. द जिनॉमिक्स इज ऑल्सो हेल्पफुल इन फोरेन्सिक एनालिसिस. You know, forensic analysis means the science which refers to the study of different aspects which is helpful to identify the crimes and criminals. Next one, the knowledge of genomics is helpful in the development of genetically modified plants, genetically modified animals and microorganisms also. Genetically modified plants and animals means transgenic plants, transgenic animals. What does it mean? We have to study separately. But temporarily I want to tell you the genetically modified organisms are developed by the introduction of certain desirable gene of other organism so as to develop some desired character. जेनेटिकली मॉडिफाईड ऑर्गॅनिझम्स डेव्हलप करत असताना दुसऱ्या सजीवामध्ये असणारे कुठले तरी उपयुक्त असे जीन्स त्या सजीवात आणले जातात की ज्यामुळे त्या लिव्हिंग ऑर्गॅनिझम मध्ये डिझायर्ड कॅरेक्टर्स, मानवाला उपयुक्त गुणधर्म निर्माण होऊ शकतात. अर्थात दिस टेक्निक इज यूज्ड, दॅट इज द टेक्निक ऑफ डेव्हलपमेंट ऑफ जीएम, genetically modified organisms or transgenic organism is very useful in the development of so many crop plants, fruit plants which show maximum desired characters. Also for the development of different transgenic animals so as to obtain different desired products from them. Also this technique is useful to develop different transgenic or genetically modified microbes so as to obtain so many useful products from them such as vitamins, enzymes, antibiotics and so on. So the knowledge and understanding of genomics is highly useful for human being. So here we have discussed about the genomics. Now we have to talk about the next topic, that is human genome project. Let us move to the next. Human genome project. Dear friends, just now we have completed the study of genomics and now let us talk about the human genome project. Human genome project, it was an international comprehensive effort or it was an international collaborative effort started in year 1990 by molecular biologists of different nations to map the entire human genome by studying, by sequencing and by mapping all the genes present in DNA of chromosome present in human genome. We know that the human genome consists of haploid set of chromosome, that is 23 chromosome, of which 22 autosomes and X as well as Y chromosomes. So, in this human genome project, it was aimed to find out the entire nucleotide sequence, to find out the location, as this human genome project was an international collaborative effort. Therefore, the molecular biologists of so many different nations contributed a lot in this project. The international partners were USA, UK, that is United Kingdom, France, Germany, Japan, China, as well as India. But this entire human genome project, it was initiated and led by the US Department of Energy and National Institute of Health, which was led by Francis Collins and Craiz. So I was telling you, the Human Genome Project, it was an international collaborative, international comprehensive effort. It was initiated by the US Department of Energy and National Institute of Health in year 1990, and though it was an international effort, it was led by the molecular biologists such as the Collins and Craiz. It was expected to complete this entire project within 15 years, and the estimated cost was $3 billion. Though it was decided, it was estimated to complete this project within 15 years, but major part of this human genome project get completed just within 13 years, that is in year 2003. The major draft of human genome project was ready. Of course, the credit goes to the hard efforts taken by the molecular biologists as well as the computing system, because whatever huge data obtained by the scientists while performing this work, that was analyzed, properly stored with the help of computing system. Now, what were the aims of Human Genome Project? Let us talk. First, mapping of entire human genome by studying the nucleotide sequence of DNA of all chromosomes present in human genome. As I already told you, that is 22 autosomes as well as X and Y chromosome. Second aim, to store this entire data, to store this entire information obtained from the Human Genome Project in the form of databases, which is necessary for its further use as well as for its further analysis. Third aim, to find the functional role of all the genes. To find functional role of all the genes present in human genome, as well as to take the care of different social, ethical issues which may arise during the study of human genome during the Human Genome Project. So, the major aim of Human Genome Project was the complete analysis of human genome to map and sequence, that is mapping and sequencing of entire human genome, as well as to find the functional role of genes present in human genome. Now, through this human genome, so many different facts came into light, but simply here we have to study some of the important highlights of the Human Genome Project. So let us talk about that highlights. Here I have written some of the major highlights of the Human Genome Project. Let us talk one by one each. First, during Human Genome Project, two types of maps were generated. During Human Genome Project, two types of maps were generated, such as the genetic linkage map and physical map. Now here we don't have to discuss what does it mean, but physical map, it refers to the mapping, that is to find out the actual locus of genes on the chromosome, and linkage map, it describes the position of different linkage groups on different chromosome. We people have already studied about the linkage, types of linkage, subtypes of linkage. We know that linkage, it is the tendency of genes present on the same chromosome to remain together and inherit together, and that genes which are present on the same chromosome form one linkage group. So the exact number, location of such linkage groups is determined here in genetic linkage map. So the first highlight I told you, during Human Genome Project, two types of maps were generated, such as genetic linkage map and second physical map. Human genome consists of 33,000 genes instead of 80,000 or 1 lakh genes which were expected before the beginning of Human Genome Project. Means initially it was expected that there should be near about 80,000 to 1 lakh genes in the human genome. अर्थात सुरुवातीला असं एक मानलं जात होतं की सर्वसाधारण ह्यूमन जिनोम मध्ये 80,000 to 1 lakh genes असावेत. But after the Human Genome Project, it came to know that the human genome consists of only 33,000 genes, and the C value of human genome is 3 billion. Genome तयार करणाऱ्या DNA मध्ये एकूण न्यूक्लियोटाइड पेयर्सची किंवा नायट्रोजन बेस पेयर्सची संख्या किती आहे याला म्हटलं जातं C value. And here the human genome consists of about 3 billion base pairs. Therefore, the C value is 3 billion. The human genome draft represents about 90% of genome. Please try to understand. I initially told you it was expected to complete the Human Genome Project within 15 years. It was initiated in year 1990 and was expected to complete up to year 2005. But about 90% of the draft was get prepared within 13 years, that is in year 2003, and the 10% part remained with less important genes. It means whatever human genome draft proposed or put forth by the scientists that were with the detailed mapping sequencing of 90% of the genome, and 10% part remained with less important genes which are present near the telomere of chromosome and around the centromere of chromosome. So here the third important highlight is that the human genome draft represents about 90% of the genome and 10% of the genome remain with less important genes. Also, it was found that all humans are alike genetically by 99.9%. याचा अर्थ genetic level ला all human beings, they are alike, they are identical by about 99.9%. and the variation seen among human beings is only due to the difference in 0.1 to 0.2% of the genetic material. ह्यूमन बीइंग्स मध्ये दिसणारं जे व्हॅरिएशन आहे, विविधता आहे, ती केवळ फक्त 0.1 to 0.2% मध्ये असणाऱ्या जेनेटिक मटेरियल मधल्या फरकामुळे, डिफरन्समुळे आहे. The DNA molecule we have already studied shows some coding sequences. Coding sequences means the sequences, nucleotide sequences which contain information, basic genetic information for polypeptide or protein molecule, and some non-coding sequences means the nucleotide sequences which does not possess information for protein or polypeptide synthesis. We know that the coding sequences are also called as the exons and non-coding sequences are called introns. It means the coding sequences or exons are interrupted by means of introns, and these introns, these non-coding sequences, they show repeatedly occurring short sequences of nucleotides. Let me explain you. Suppose, consider, here is the DNA molecule. In this DNA molecule, these are exons or coding sequences, and these are introns, that is non-coding sequences. Just consider, these pink colored are supposed to be the introns and yellow colored exons. I already told you that exon, these are coding sequences, and intron non-coding. I said that the intron, suppose this is one intron. Intron shows repeatedly occurring short sequences of nucleotides. Single intron shows repeatedly occurring short sequences of nucleotides. Please look at the diagram. Suppose here is the first base pair G, C, T, A, T, A, T, C, G. Now here I have drawn only a short nucleotide sequence of 5 base pairs. Now this short nucleotide sequence occur repeatedly in one intron. Means again the same nucleotide sequence is present here in that intron. Same G, T, A, C. Again G, T, A, C. In this manner. Of course, the number of these repeatedly occurring short sequence of nucleotide is different. प्रत्येक मानवामध्ये ह्या ज्या शॉर्ट सिक्वेन्सेस आहेत न्यूक्लियोटाइड्सच्या, त्यांची संख्या इंट्रॉन्स मध्ये वेगवेगळी असू शकते. And such repeatedly occurring short sequence of nucleotides in the intron is called as VNTR. VNTR, that is variable number of tandem repeats. This VNTR has great importance in the DNA fingerprinting, about which we shall study in next topic. So I said that the DNA molecule consists of coding sequences and non-coding sequences. Coding sequences are called exon, non-coding sequences are called intron, and this intron they show VNTR, that is the repeatedly occurring short sequence of nucleotides. Now the next highlight, through the Human Genome Project, it was found that the insulin gene and beta globulin gene are the shortest genes in human genome. Shortest genes having the size of only 10 kilo base pairs. 10 kilo base pairs. You know base pair, here is one base pair, GC. 1 kilo base pair, it means 1000 base pairs. 1000 base pairs is equal to 1 kilo base pair. And the insulin gene and beta globulin gene are the shortest and consist of 10 kilo base pairs. While the longest gene is a dystrophin gene. Dystrophin gene. This gene is responsible to cause disorder, that is muscular dystrophy, Duchenne muscular dystrophy, and this dystrophin gene is the longest gene and it is made up of 2400 kilo base pairs. Dystrophin gene is made up of 2400 kilo base pairs. Another highlight, according to the Human Genome Project, the first chromosome in the Human Genome Project is longest one. We know that the human genome consists of 23 chromosomes, that is 22 plus X or 22 plus Y. So out of these 23 chromosomes, the first chromosome, first autosome is longest. It shows about 2968 genes. 2968 genes. While the Y chromosome is shortest one and it contains very few genes. Also, it was found in human genome there are presence of about 1200 genes which are responsible to cause different disorders, different diseases such as breast cancer, cardiovascular diseases, color blindness, diabetes, Alzheimer's disease, and so many. So here we have considered some of the important highlights of the Human Genome Project. In this manner, we have completed the study of two topics here, such as genomics and Human Genome Project. So let us stop here today. Thank you.